Hello and welcome to Bio40 Pre-Lecture number 6, a continuation on the chromosomal basis of inheritance. We ended last class talking about mitosis and meiosis. Observing the movements of chromosomes during these processes was key to the theory that genes exist on chromosomes. For this theory to work, the ways traits are transmitted should correlate to how chromosomes are transmitted. Several of the qualities of chromosomes that we discussed last time line up quite well with the qualities of genes. Most cells have homologous pairs of chromosomes, just as they should have two alleles of each gene. The main exception is the gametes, which are haploid, and indeed should have only one allele. Genes should be the same from cell to cell, and mitosis involves each daughter cell having identical chromosomes. Mendel's first law of segregation is mirrored by how homologous chromosomes pair and separate in meiosis I, and Mendel's second law is reflected in how the segregation is random in relation to maternal and paternal chromosomes. Let's go into a little more depth about that last point. Suppose the yellow and blue chromosomes are a homologous pair, as are the green and the red. This individual inherited the chromosomes on the left from her father, and the chromosomes on the right from her mother. When they separate in meiosis I, the two paternal chromosomes and two maternal chromosomes could stay together, or they could segregate such that each cell gets one maternal and one paternal chromosome. The non-homologous chromosomes group randomly. So for a species with a haploid number of n, there are 2 to the n potential groupings of chromosomes. And that number isn't even considering how crossing over can further increase the potential combinations of alleles. That last slide explains independent assortment of alleles on different chromosomes. But a chromosome cannot just be a gene, as humans probably have more than 23 traits controlled by genes. So a chromosome must contain many genes. If that's the case, how can they assort independently? Well, suppose genes A and B are on the same chromosome as shown here. Someone inherits a dominant allele of each gene from one parent, and a recessive copy of each gene from the other. Without crossing over, you can never have a gamete that's dominant for one gene and recessive for the other. But when portions of sister chromatids exchange in crossing over, you can get all 4 combinations of alleles predicted in independent assortment. So we've found a good correlation between the transmission of genes and the transmission of chromosomes. But to really establish the link between the two, we want to show that specific traits are associated with specific chromosomes. Thomas Hunt Morgan did this using a sex-linked trait for white eyes in Drosophila. Typically, fruit flies have red eyes. This is a wild type. Morgan bred flies until he found a mutant phenotype that would be easy to study, and eventually found a white-eyed male. When he crossed this fly with a wild-type female, all their offspring had red eyes. This should be sufficient to tell you the white eye allele is recessive. When he crossed the F1 siblings, he found a 3-to-1 ratio of red and white-eyed flies, again consistent with the recessive trait. However, analysis by sex showed that all the females had red eyes, and the males were evenly split between red and white. Morgan concluded the eye color gene was on the X chromosome. So the parents were heterozygous and hemizygous. The F2 females were evenly split between homozygous dominant and heterozygous, and the F2 males were all hemizygous, but with an equal chance of having a red or white allele. Note how the alleles are displayed here. Genes on the X chromosome are shown as an X with the allele indicated in the superscript. W+ is used for dominant wild-type alleles, and W's without pluses are recessive mutant alleles. Morgan supported his model with further crosses. He saw that mating heterozygous females and white-eyed males gave an equal mix of red and white-eyed offspring amongst both sexes. See if you can tell what their genotypes are. Morgan also crossed white-eyed females with red-eyed males. This time he got only red females and white males. This is called crisscross inheritance. The daughters had the phenotype of the fathers, and the sons had the phenotype of the mothers. This is a hallmark of sex-linked inheritance. Another hallmark of X-linked inheritance is getting different outcomes from reciprocal crosses. You get different offspring crossing a red female and white male than when you cross a white female and red male. So Morgan saw all these telltale signs that the white-eyed trait was transmitted with the X chromosome. But the best evidence came from Morgan's student, Calvin Bridges. Bridges scaled up the number of flies he looked at and saw that 1 in 2,000 offspring of the crisscross mating violated the typical pattern. These were white-eyed females and red-eyed males. And for some reason, all such males were sterile. What could explain this? Bridges proposed a rare event called non-disjunction. In such cases, when the females form their gametes, instead of each gamete getting one X chromosome, one gamete gets two X chromosomes and one gets none. Let's look at a Punnett square of these gametes combining with typical male gametes. There are 4 possible outcomes, but flies with 0 or 3 X chromosomes are never born and thus not observed. Of the flies that live, half will have a Y chromosome and 2 X chromosomes with white alleles. These will be the white-eyed females. The other half will have just 1 X chromosome with a red allele. These will be red-eyed males, and the lack of Y chromosome explains the sterility. Bridges confirmed this was the case cytologically, directly observing the abnormal configurations of chromosomes. Now at this point, I'm pretty convinced the eye color gene is associated with the X chromosome, but Bridges didn't stop here. He took the fertile XXY females and crossed them with wild-type red-eyed males. And he found some more peculiar results that even further supported the relationship between genes and chromosomes. But let's save that for class. For now, we'll take a closer look at how non-disjunction can occur in meiosis. It can actually occur in meiosis 1 or 2. In meiosis 1, it involves the entire tetrad of homologous chromosomes moving into one of the daughter cells. So after meiosis 2, 2 gametes have extra X chromosomes and 2 have none. Non-disjunction in meiosis 2 means both daughter chromosomes move to the same daughter cell. One gamete has 2 X chromosomes and one has 0, but the other 2 gametes are normal. Now let's look at the other interesting aspect of the cross. How is the sex of the flies with odd numbers of sex chromosomes determined? This is actually very different in flies and humans. In terms of sex assigned at birth, XX individuals are typical females, and XY are typical males for both people and flies. But XXY flies are normal females, while XXY people are sterile males, with other atypical phenotypes. Meanwhile, XO flies are sterile males, while XO humans are sterile females, also with additional phenotypes. This is because it's the presence or absence of the Y chromosome that determines sex in humans, while the ratio of autosomes to X chromosomes determines sex in Drosophila. Flies with 2 X chromosomes and a normal diploid set of autosomes have an X to autosome ratio of 1 and are female. Diploid flies with 1 X chromosome have an X to autosome ratio of 0.5 and are male. This explains the XXY females and XO males in Bridges' cross. Now I want to move back to humans. In people, autosomal aneuploidies, that is having an abnormal number of chromosomes, are mostly lethal, and are, at the very least, highly detrimental. But here we have 4 cases with abnormal numbers of sex chromosomes that have only small or nearly no detrimental effects. If aneuploidy is normally such a problem, why can we tolerate anywhere from 1 to 3 X chromosomes? And why can't flies tolerate the same? The answer involves dosage compensation. Humans and flies must be able to tolerate varying numbers of sex chromosomes, because even in typical cases, males have 1 X chromosome and females have 2. In humans and other mammals, this problem is solved by X inactivation. In women, 1 X chromosome is inactivated, forming what is called a bar body. Around the 16th day of human development, when there are around 500 to 1,000 cells, 1 X chromosome in each cell inactivates. It's random which one is inactivated in each cell, but it's irreversible from this point. All descendants of a given cell will have the same inactive X chromosome. The chromosome will only reactivate in germ cells. Women who are heterozygous for genes on the X chromosome will have different active alleles in different cells, and are therefore genetic mosaics. This mosaicism can easily be seen in calico cats. The gene that controls black versus orange fur is on the X chromosome. They have black patches where the orange allele is inactivated, and orange patches where the black allele is inactivated. The same phenomenon can be seen in the human condition, anhydrotic ectodermal dysplasia. This is a sex-linked recessive condition leading to several phenotypes, including a lack of sweat glands. Heterozygous women are mostly unaffected, but if you examine their skin closely, you'll find there are actually patches of normal skin where the disease allele is inactivated, and patches of skin lacking sweat glands where the normal allele is inactivated and only the disease allele is active. Understanding dosage compensation can be easier when you consider gene products. what the gene actually makes. For autosomes, there are normally 2 alleles, so you get 2 doses of gene product. In monosomy, there's only 1 chromosome and 1 dose, while in trisomy, you have 3 copies and get 3 doses. Cells are programmed to tolerate exactly 2 doses of autosome gene product. So monosomy and trisomy are generally not tolerated in autosomes with no dosage compensation. In humans, either 1 or 2 X chromosomes are to be expected, depending on sex, and we have dosage compensation by X inactivation. Our cells are set to tolerate 1 dose of gene product from X chromosomes. So when we have 3 X chromosomes, we just inactivate 2 of them and still get 1 dose of gene product, which is just fine. But fly dosage compensation works differently. In Drosophila, males with only 1 X chromosome are compensated by hyperactivation. That is, their single chromosome produces 2 doses of gene product to equal what the females make with 2 X chromosomes. Since flies can only compensate by increasing gene product, there's nothing that can be done to make up for the extra dose produced by having 3 X chromosomes, so such flies are not viable. So if X inactivation deals with sex chromosome aneuploidy in humans, why do XXX, XXY, or XO individuals have any atypical phenotypes at all? The answer is that X inactivation isn't quite complete. The ends of the X and Y chromosomes have pseudo-autosomal regions, par1 and par2. These regions are important in allowing the X and Y chromosomes to pair during meiosis. When an X chromosome is inactivated, the genes in par1 and par2 remain active. Cells still need 2 doses of these genes to function normally, so XXX and XXY individuals still have extra doses of these genes, and XO individuals don't have enough. Additionally, some X genes that aren't in par1 or 2 are not fully inactivated. This all is enough to cause some problems in individuals with sex chromosome aneuploidy, but is not enough to be lethal. Okay then. This all is sufficient to establish that genes are on chromosomes. But what does it really mean to say that a gene is on a chromosome? Next time, we start mapping genes to specific locations along the chromosomes.